Positron emission tomography diagnosis, which is commonly known as PET, is used worldwide as an advanced method for diagnosing cancer. Taiyo Nippon Sanso is the world's largest oxygen isotope manufacturer and supplies water 18O, oxygen 18 stable isotope labeled water, which is the starting material for PET diagnostic agents, to over 40 countries. In 2004, we put the world's first oxygen distillation plant into operation with the uniquely developed cryogenic distillation technology and realized the world's highest level of isotope concentration at 98 atom%. With a subsequent increase in demand, we built our second plant in Soregaura City in 2013 and a third plant in Shunan City in 2016, establishing a stable supply system with an annual production capacity of over 600 kilograms across these three independent plants. With regard to purification, sterilization, analysis, and other final processes, a high-standard quality control system has been established at the SI Innovation Center in Thomas City. PET diagnosis employs the measurement and mapping of gamma radiation that is emitted when a positron, which is released from radioactive drugs, binds with an electron. It converts the water 18O starting material to 18F through a nuclear reaction. This creates the diagnostic agent 18F-FDG. It helps in early cancer detection, identifying the location and extent of a cancer, assessing treatment efficacy, and diagnosing reoccurrences and metastases. In recent years, PET diagnosis has begun to be used as a diagnostic method for Alzheimer's disease and myocardial blood flow measurement. We will support PET diagnosis through the stable supply of water 18O, contributing to the health of everyone. Deuterium is a hydrogen isotope. The hydrogen atom H consists of one proton, which has a positive charge, and one electron, which has a negative charge and is much lighter than the proton. On the other hand, the deuterium atom D consists of one neutron, which has a mass roughly similar to that of a proton and no electric charge, in addition to one proton and one electron. For this reason, the mass number of deuterium is about twice that of hydrogen. Furthermore, tritium, another hydrogen isotope, has two neutrons, so the mass number is about 3 times that of hydrogen. A stable isotope, deuterium exists in very small amounts in nature. The mass of deuterium is greater than that of hydrogen, and deuterium bonds more strongly with carbon and oxygen than hydrogen. This isotope effect makes the molecule more durable and can improve the performance of various functional materials. In organic electroluminescence applications, for example, the durability and light emission efficiency of the element can be improved. In semiconductors, for example, circuit durability can be improved, and in pharmaceuticals, long-lasting medicinal effects can be obtained. Taiyo Nippon Sanso synthesizes various deuterium compounds. One of these deuterium compounds, deuterated ammonia, is a stable isotope gas for which Taiyo Nippon Sanso established a mass production system for the first time in Japan. We will respond to the increasing demand for its use in semiconductors as a chemical reaction raw material and in analysis. Deuterium-rich water, commonly known as heavy water, is essential in fields like nuclear energy and NMR research. It also provides deuterium for creating a wide range of valuable compounds, supporting scientific innovation. Domestic demand for heavy water is entirely dependent on imports from overseas and vulnerable to overseas supply and demand conditions, as well as the export policies of manufacturing countries. Stable procurement of heavy water is not easy. Taiyo Nippon Sanso has developed equipment for re-enriching used heavy water to establish a heavy water recycling system. This enables efficient and sustainable use of heavy water and contributes to the stable supply of deuterium compounds to meet domestic and international demand. Cell-free protein synthesis is a technology that enables efficient in vitro protein production using the essential components extracted from living cells. Compared to conventional culture-based methods, it can synthesize a wider variety of products, it offers high throughput, and it has a higher success rate in protein synthesis. So, how are proteins actually made? Proteins are typically produced inside cells. The genome, which is a collection of DNA, contains all the genetic information. From this DNA, a blueprint for each protein is created. This is RNA polymerase. It reads the DNA sequence and assembles the corresponding RNA bases to match. This is a process called transcription. The resulting strand, known as messenger RNA, serves as the blueprint for building proteins. Next, ribosomes, the protein assembly factories, bind to the messenger RNA and begin reading its instructions. The amino acids specified in the blueprint are delivered by transfer RNA. Each transfer RNA carries a set of 3 bases called an anticodon, which perfectly matches 3 bases on the messenger RNA. This ensures the correct amino acids are added in sequence, building the protein step by step. This process is known as translation. The resulting long chain of amino acids folds into a unique 3-dimensional shape, becoming a functional protein. Each protein is crafted according to instructions from DNA and goes on to perform a wide variety of roles within living organisms. The cell-free protein expression kit, Musaibokun, harnesses the protein-making machinery from cells, allowing users to produce proteins anywhere, in a test tube, without the need for living cells. Musaibokun is a kit product of Riken's advanced cell-free protein synthesis technology. It offers high isotope labeling efficiency and enables rapid, large-scale protein production with ease, thanks to the use of E. coli extracts. It can also produce proteins similar to antigens and antibodies, as well as biologically active proteins and enzymes. In addition, it allows for the flexible design and creation of entirely new novel proteins. By combining cell amino acids produced with advanced organic synthesis technology with Musaibokun, users can easily create cell-labeled proteins. Detailed structural information about proteins can be obtained by NMR analysis on these cell-labeled proteins. This technology plays a vital role in advanced research fields such as drug development and the study of cancer mechanisms. The SI division will continue to support the advancement of science, technology, and people's lives through its ongoing work with stable isotopes.